Hello everybody, my name is Olaf Schmidt and I work in the UCD School of Agriculture and Food Science, where I teach students in agricultural sciences, agri-environmental sciences, forestry and horticulture. My main field of expertise is soil ecology, so they're all the living things in the soil and their functions. And I'm happy to share with you a little field video that we have produced recently on how to sample earthworms and how to measure soil density. Hello everybody, you're very welcome to Soil Science Applications. My name is Olaf and I'm here with Laura, and at the moment we talk about soil as a living entity and soil biology, and it is an awful shame that you can't be here for this field practical, but we will do our very best to show you what this is all about and what you're supposed to learn. Okay, let's do this. So this afternoon we are in Rosemount Environmental Research Station, which is on the UCD campus. It is a lovely facility. There are greenhouses, there is an orchard, there are beehives, and there also is a small lab inside where we can usually take students. And today we are here because we just need a small piece of land, a lawn where we will sample earthworms. Just to introduce the scientific content, as you recall, in my lectures we have learned about the biology of soils and the biodiversity. There is a huge biodiversity of all the different taxa, so there are soil animals, microbes, bacteria, fungi and so on. And as an example or as a case study, we have selected earthworms because they are just really nice to see and to study. You can, sample them yourself, you can see the different species, and also you can see the activities. Okay, so all of this we will now do in this field practical. What we want to do this afternoon is to sample earthworms and extract them from the soil. We will be using a dynamic method or a behavioral method that exploits the escape behavior of the earthworm. So in essence, you irrigate the soil with a liquid irritant, it infiltrates, it hits the worms, and the earthworms will surface where you collect them. Just to start off with, here is all the equipment that you need. So in this case we use a 50 by 50 centimeter quadrant to define your sampling area. You need a watering can, you need shears to clear the grass so that you can actually see the soil surface and the earthworms. You need a wide-mouthed plastic sampling bottle, forceps, you need gloves of course to protect your hands, and you need the magic ingredient, which in this case is a mustard oil concentrate. So this is a spicy irritant that you mix in with the water. You also need then in the lab a tray where you can sort the earthworms into the ecological groups, and you might need a magnifying glass. What you also do, you need a soil thermometer, because this is a behavioral method and the earthworms have to be active in order to actively be expelled from the soil. So the first step is that you decide where to sample. In this case it's 50 by 50 centimeters, and you only sample the worms which emerge in that area. So later on you can upscale that and multiply it up to, you know, a square meter or a hectare. You chop off the vegetation. Don't walk on the land, because the earthworms will detect vibrations and will escape downwards and not upwards. And then you need to mix the irritant liquid. So as I said, this is a liquid made from diluted mustard oil, and the active ingredient is actually only 2 millimeters in 20 liters. Okay, if you would like to do this at home, just use a very spicy eating mustard and make a suspension, and it will have the same effect. Okay, so we will do this now, and then we will show you how to extract the worms. Okay, so I put that in. Okay. So here is our sampling spot. We have cleared the vegetation so that we can see the soil surface. Beforehand, we have measured the temperature of the soil. It is 12 degrees at the moment, which is really good. So for these behavioral methods, you want the soil temperature of at least 10 degrees, okay, because the earthworms have to be active and mobile. We will now apply the irritant and see what happens. So you need, you know, a watering can with a rose, and you apply it evenly. You will need in the order of 5 liters. Okay, now it will depend on the type of soil and the moisture content of the soil. Okay, so the principle of this method is that the irritant will infiltrate in the soil into the earthworm channels. It'll hit the earthworms, which will feel agitated and irritated, and they will escape upwards and you can pick them up. Okay, so you can see now it starts to work. I don't know if you see this. Here's the first earthworm. It's a bit too small to see. There's the second one. So hopefully we will also have some larger ones. Do you see that here? A very large one. Do you see? So that's our first worm. Okay, and you put that in your sampling bottle. You have to wait until all the worms have completely left the soil. Okay, so in particular these Anisix worms, the big species, as you saw in the lecture, they will stay with their tail in their channel. They have setae or bristles with which they fix themselves in the soil, and they think you want to eat them, or you are, you know, a predator, and you will not be able to pull them out. Okay, so you have to wait. Do you see this one here in the middle? So you do have to wait, okay, until they have left the soil, and then you can pick them up. You've maybe noticed we actually waited a little bit for the sun to go away, and it's now perfect because it is overcast, because earthworms hate the mustard oil, but they also hate, you know, the sunlight, because the UV fraction will kill them. So they find themselves between a rock and a hard place. So this method will work better if it is overcast as it is now. Okay, so we start to see the first ones to emerge. They are a little bit slow, so I will apply more solution now. So you do it nice and slowly, so you have no runoff of that stuff and it stays within your square. Okay, and you have to wait for, you know, maybe 15 minutes or so, because if you think about it, some of these worms will be a bit deeper in the soil. They also, some of them have horizontal channels and they actually have no opening at the soil surface, so you need to give them time to work their way out. Here's a nice one. So you see the equipment we are using in the field. Never use glass bottles, okay, because they can break. Always use plastic ones, which you can also easily label. Okay. So we are now at the end of the extraction on this spot. We waited for about 15 minutes. We applied a second lot of the irritant, and you can see we have a reasonable number of worms. There are, you know, about 30 worms, and since this is 50 by 50 centimeters, if you multiply this by 4, you would have the earthworm abundance in the meter square. And then in the lab you could also weigh them, their biomass, and you would also multiply that. So the next step then that we will show you is that you would also separate them into the ecological groups. In order to show you the extraction even better, we set up a second plot and we produced this time lapse where the first 5 minutes are shown within 40 seconds. So you can see the 50 by 50 centimeter plot. The mustard oil liquid is applied slowly and evenly, and then within a minute or two the first earthworms are surfacing. You can see that the large worms that have openings at the soil surface will emerge first and appear at the soil surface. Okay, so the last bit then of the earthworm extraction, well, first of all you need to rinse them in tap water because you need to wash off the mustard oil off their skin or it will kill them. And then you put the worms onto a tray like this and you sort them. Okay, I don't know how well you see this, but first of all you would sort them into the juveniles and into the adults. As you recall in the lecture, adult worms have a saddle or a clitellum near the head, okay, whereas you don't see this in the juveniles. And then you would also sort them into the ecological groups. Okay, so here we will use a simple system where we will just look for small worms which are red, like this one and this one. They are the epigeics. Okay, then we look for white worms or gray worms which are not red pigmented, and these ones are the endogeic worms. And finally you can see in that sample here we have 3 adults of the anisic. So these ones are, you know, the very large bodied species which make vertical channels. And then you would count them and write that down, you know, in your notebook. If you would do this more scientifically, you could also take them in the lab and do the actual identification of the species with a hand lens or with a microscope. So I just want to show you, I don't know if you can see this. So this is a Nisic, okay. This is an adult, because here is the head end, and you can see here this bit which is orange and thicker is the saddle. Do you see that? And here's a second one which is a different species. So here again you can see the saddle here. Do you see this? So these species are different species. This one here is is a species called Abractodea longa, or the black-headed pasture worm. This is extremely widespread, in Ireland, and you would have this in most grassland soils. Whereas this species is called Lumbricus terrestris, or the dew worm, which indeed is the largest earthworm species we have in Ireland. Okay, so you can see in this patch of lawn here in Rosemount we have, in the order of 5 or 6 different species. Here is an endogeek. You can see it is much smaller, but you can also see the saddle here. Okay, if you can see this in the resolution.
No.
This is a species called Allelophophra chlorotica, and it is also very common. So this is an endogeek worm. It means that it lives in the mineral soil, eating mineral soil and making vertical channels. And this one here is a juvenile of an epigeek. You can see it has no saddle, okay, so it looks the same all along the body. And these species are usually heavily red pigmented, because living at the soil surface or in the root mat, they are at the risk of exposure to UV light. So this is why they are red pigmented. In this practical, of course, when you're done, we don't have to kill the worms. So the final thing now we do is we will let them go, but not on the spot where we use the mustard oil, because obviously they won't like that. Okay. So you've seen, the principle of sampling earthworms with a liquid irritant. You would, of course, let's say if this was a research project, you would always need several replicates. Okay, so in a piece of land like this, you would maybe do 5 or 10, so that you have, statistically sound results. The distribution of the earthworms within a landscape is, heterogeneous. So this is why you would always have replicates, and you would pick them randomly. Okay. In that last section now of the field practical, we want to measure penetration resistance of the soil. So this is a measure of the level of compaction of a soil. So if a soil is very compacted, it will take a greater force to penetrate it. And this is a handheld mechanical penetrometer. You can see it's the size of a large pen, and it is really simple. So at the end here there is a tip, and this is the bit you push in the soil steadily and evenly, and then you can see here is a rubber ring, and this unit here on the scale, if you can see this, is the force it has taken to push this in the soil. Okay, so the beauty of this instrument is that it's extremely simple, and I will now demonstrate its use on this little piece of soil. So here is a small piece that we have left uncompacted, whereas on that side I trampled on and it is compacted soil. So you take the instrument and you push it in the soil up to this measurement here, and then this will give you the reading. Okay, so this is the force in kilograms per square centimeter, and you can see that this is a uncompacted soil, whereas if you do the same on the other side, you can see you need much more force to push that in to the same depth. So you can see that this is almost twice as compacted as before. So this is a really easy method, very low tech. Of course, the disadvantage is you would have to take many measurements because each individual measurement isn't accurate. So if you would assess a piece of land, you would maybe take 20 measurements or so. I hope you will find that useful. If you have any questions or feedback, feel free to email me at olaf.schmidt@ucdie. Thank you.